Okay. So this lecture, you will learn two main group of bacteria, but two of them are spore-forming gram-positive bacteria. Spore in this term mean endospore forming. Okay. Let me give you summarize from the previous lecture. So you learn basic bacteriology already. Then we move to gram-positive. Okay. Gram-positive cocci are done in lecture 12, Staphylococcus, lecture 13, Streptococcus, and Enterococci. Okay, so main thing, you need to go back and identify the major characteristic of those bacteria. What are the common characteristics that all Staphylococcus should be, all Streptococcus should be, and how different between Staphylococcus and Streptococcus. Please not tell me like arrange in cluster and arrange in chain, that's it. So you learn more the way they cause disease, some biochemical reactions, drug susceptibility, something like that. And then we move to gram-positive bacilli. Gram-positive bacilli, we divide to be non-spore forming and spore forming, right? Still remember? Non-spore forming gram-positive bacilli separate by the way of staining and the pattern of morphology. If they have regular morphology, regular gram staining, then we put in one group. If they have irregular, I mean impossible to stain by gram or have weird shape, not just regular bacilli, they form the club shape, they have filamentous, then they put aside in a group of irregular. But now, this lecture, we talk about endospore forming. In the whole kingdom of bacteria, as long as I review, there are three, well, if they reclassification, maybe the new thing, three genus of bacteria, three genera of bacteria that can produce endospore. One is Sporosacina. Sporosacina is non-pathogen. So in this course, if you don't know something about Sporosacina, that's fine. The second one, the name probably familiar, is Bacillus. Okay, the Bacillus. The third one is Clostridium. I think most of you know this Bacillus and Clostridium already. The key of this group of bacteria are food poisoning, okay, because they contaminate in food and they can do something else also. Let's start from the endospore formation. So endospore, like I said in the last week, endospore is not a process that bacteria produce for reproduction, but endospore produced for bacteria to tolerate the harmful environment. Okay. Normally, if you see this cycle, normally bacteria can produce another cells by binary fission. One bacteria generate another small duplicate chromosome and then duplicate so many protein inside mRNA, rRNA, and both of them split into two compartments by cell membrane. Then cell wall break up. If you learn genetic, microbial genetic or molecular biology, you probably heard the term competent cell. Have you know about this term? What is competent
yes.
cell? Who want to try what is the competent cells? When you culture E. coli or Bacillus, then bacillus turn to be competent. Competent is what?
They can readily take up plasmid.
Plasmid. And how the cells look like?
The cell walls are slightly thinner, I think,
Thinner? What
or
thinner?
porous.
Porous?
There's
What part
yeah,
of the cells get porous?
there's cell membrane, cell wall.
The membrane cannot get porous, otherwise they die.
Okay,
Okay. Cell
then
wall. You
cell
imagine
wall, yeah.
eggshell. If the egg, if just imagine, if egg want to be grow up, the shell need to be break. And then once the shell break, cells elongate. Eggshells move from the pole, and then the new shell polymerize and then seal. Then rigid structure can move up, right? At this time, cells are so fragile because cell wall break up. So the new cell wall need to be synthesized so quick. Okay, that's the thing. We spend antibiotic to attack bacteria. If the cell not growing, not dividing, antibiotic cannot inhibit bacteria. Okay, so antibiotic will act well when bacteria grow. In case of harmful environment, bacteria produce another set of chromosomes and put the chromosome in the compartment. They form a membrane structure compartment around new chromosome. Okay. The big compartment, that will be a mother cells. The small compartment is pre-endospore, okay, or pre-spore. They are pre-spore membrane, 2 or 3 layers that have been produced. Finally, chromosome of mother cell has been digested. Once chromosome of mother cell digests, protein digests, endospore remain inside the cell. Okay, but cells die. Cell cannot have any function more. Endospore kind of pack. Okay, once the cell lysis, mother cell lysis, so mother cell die completely. Endospore can tolerate to the harm environment. Endospores of some bacteria can stay in environment almost 100 years, and they can reach and break out the spore. We call kind of germination out, but germination get one bacteria. Okay, that's the cycle of endospore. So well, when we talk about endospore formation, that bacteria need to be gram-positive bacilli. The other shape of bacteria cannot produce spore except bacilli. We separate spore-forming bacteria by the ability to consume oxygen of bacteria. One side of them are, one side is aerobic bacteria. Another side is anaerobic bacteria. For aerobic bacteria, we point to the genus Bacillus. For anaerobic bacteria, we point to the genus Clostridium. Okay. Genus Bacillus is very huge. Genus Clostridium is also huge. Okay. But even they contain like more than several hundred species of bacillus, but just only 2 bacillus cause disease with human. I will not go to detail of anything in this lecture. I try to reduce the number that you need to remember, but just concept, okay? Let's start from genus Bacillus. This is picture of Bacillus. So Bacillus is relatively large bacilli cell. Lots of species arrange the cell in chain. You probably see the bacillus linked together like a long chain, okay, like a train. Some bacillus can move, okay, some cannot. Um, Even the bacillus contain like 100 species, but genetic material inside bacillus of 100 species kind of very closely related, okay? But they make the phenotypic not just refer to only morphology, but metabolism, structure, or protein. They are so diverse, okay? And they cause pathogenesis are so diverse also. So genetic similar, but phenotypic and pathogenesis quite diverse. Uh, the virulence factor of these species came from plasmid, okay? Because bacillus absorb so many plasmid from nature, from the other bacteria, so they have a lot of machinery. Bacillus mostly are saprophyte. The saprophyte means they grow on the dead animal, dead plant, okay? So they can be found in everywhere. The soil, the water, in air, on the plantation area, agricultural, that's contain a lot of bacillus. Some species of bacillus are insect pathogen. For example, Bacillus thuringiensis. Let me talk about Bacillus thuringiensis a little bit. Actually, this bacillus not attack human, okay? But they attack insect. Most of insect can be infected by bacillus. B. thuringiensis produce alpha toxin. The alpha toxin is exotoxin, okay? When bacillus produce a lot of toxin inside the cells, toxin will dense in the cells and then they form a crystal protein. That's dense of alpha toxin. Once insect ingests the bacillus or the food that contain the bacillus die. Alpha toxin that is so tightly packed meet the pH in the insect gut. That is around 5. Then the toxin kind of release itself from the pack and then action kill insects. So insect die. We know the alpha toxin is came from a gene. So human clone this gene and make GMO in so many plants. So when plant produce alpha toxin, insect cannot eat. If they eat the plant that GMO for toxin in plant, bacterial toxin in plant will go to the insect and insect die. That's the GMO, okay? That's the application of Bacillus thuringiensis. There are so many research. In 30 years ago, we kind of, this bacteria is very popular. So two important pathogens for human is one, Bacillus cereus. Another one is Bacillus anthracis, okay? Let's start from cereus. The name cereus come from rice, okay? Let's see the colony of Bacillus cereus on blood agar, okay? This is a large colony. Bacillus cereus compared to all bacteria you learned before in gram-positive, they produce tiny colony, but cereus is so large. They produce beta hemolysis, okay? Most strain can be hemolysis, but there are some variety of cereus that cannot hemolyze, okay? This is very common in the air. So you can open the plate and then trap Bacillus cereus so many everywhere. Usual method of the cleaning antiseptic disinfectant is not work for Bacillus cereus. They tolerate that because they produce endospore. Even cell die, but spore remain. But if you let the time pass, Bacillus cereus can hatching from spore. That's mean regular method is not work for Bacillus group. Okay, they grow in food uh, in two ways. Bacillus contaminate the cell in the food, or actually Bacillus die, but spore contaminate in the food. So two different way. Okay, they probably mostly contaminate in the carbohydrate-rich food, for example, rice, pasta, noodle, something like this. Uh, when you, well, particularly you guys in the teenager age, you love to go to supermarket, 7-Eleven, and buy some frozen food or cook food already in the frozen, and then you use microwave, heat it. But if you not heat it properly, if that food is contaminated with cereals, so you might get infected. We, that because of the spore contaminated in the food, and particularly some students kind of super lazy. When you put the food in refrigerator and when you super hungry, you open the refrigerator and eat it cold and not heating, or if you heat but not put the right temperature, not heat it properly, that also cannot kill Bacillus cereus. Okay. Spore, not only spore, Bacillus cereus itself produce toxin, and the toxin kind of involving with, well, mainly food poisoning. Well, you might get nausea and vomiting, abdominal cramp, diarrhea. Okay. We have no treatment of Bacillus cereus, but if you get infected with Bacillus cereus, so good way to deal with Bacillus cereus is drink mineral water. And this bacteria kind of serious in immunocompromised and increase the rate of seriously every year. Okay, I think because of lifestyle of people nowadays. So you not cook at home, you kind of buy and cook. The foodborne illness, this is the disease that caused by Bacillus cereus. So we call reheated rice syndrome. Okay. Mode of transmission, like I said, they contaminate by food, by itself or endospore. They cause disease by emetic type or diarrhea type because of toxin. We call emetic toxin and diarrhea toxin. Okay. Emetic toxin happen by Bacillus itself contaminate in rice, in pasta. So when Bacillus vegetative cell contaminate, they produce toxin very quick. So incubation period disease happen within 1 to 5 hours. If you have lunch today, this evening, you probably get sick. Okay. But diarrheal type, because of contamination of spore, then you spend longer time. Okay. You might get watery diarrhea. That's mean, well, when I say watery diarrhea, that means you get diarrhea, but no blood contamination in the diarrhea. Okay. If you have blood inside that, we call bloody diarrhea. You learn diarrhea a lot next week. Okay. So watery diarrhea for Bacillus cereus. Okay. And treatment, just rehydration salt. That's it. no antibiotic use, just only for severe diarrhea. And that's supposed to be in immunocompromised, so clindamycin and aminoglycoside can be used for treatment. Okay. And this is the summary, and you can go to study yourself. Okay. So then move to another bacillus. It is subtilis. Oh, sorry, anthracis. The anthracis, you see the cells, the cells when you stain by Gram technique, you probably see endospore in the middle of the cell, and cell arranged in chain, and also block shape, is not like a curvy rod at the pole of the cells, but it's look like a block, a rectangular. Okay. On blood agar, anthracis cannot hemolyze blood. But if you observe by a microscopic of the age of colony, this area, you probably see the filament something like this a lot. We call this pattern is medusa head. This is the regular pattern of Bacillus anthracis. Okay, central spore and medusa head is the key for this bacillus. Anthracis, it is bacteria that produce capsule, but capsule of anthracis is different from the other capsule you learn. The other capsule like strep pneumo or the other bacteria, they produce polysaccharide capsule. Well, it might contain some protein, but most of the component are polysaccharide, okay? But for this anthracis, they produce polypeptide capsule. But polypeptide has just only one antigenic type, unlike the other bacteria that you know, like you get type A, type B, type C. But for this bacteria, just one type, okay? Of course, Bacillus anthracis cause anthrax, okay? Anthrax can be cutaneous anthrax, infect skin, okay? And can be pulmonary anthrax, infect respiratory tract. Also infect the GI tract. We call gastrointestinal anthrax. That's because you get contaminated spore by ingestion. The pulmonary anthrax you get by inhale inside, and then localize in the throat and also the respiratory tract, bronchi, trachea, bronchi. Anthrax itself sensitive to penicillin, but if you culture bacillus and put the penicillin, you will get a clear zone like this. This is very obvious, okay? Bacillus anthracis is known to be biological weapon, okay? Well, if you heard about the situation after 9/11 in the US, so people scare of the envelope with powder of anthrax spore, right? So Bacillus anthracis is very, very harmful for developed to be weapon because they tolerate, they produce spore, they tolerate. And when you deliver the microbe in the long way, so they not die easily. If you apply the weapon in the dry area, they tolerate that because of endospore. So, but actually produce endospore biological weapon like that is not easy as people think, okay? Another group of bacteria in the spore formation is Clostridium, okay? Genus Clostridium is gram-positive spore formation bacilli. This group is obligate and arrow. it means they cannot tolerate oxygen. If they meet oxygen, they die. But if they survive just only in the spore form, okay, this group process cannot process catalase enzyme, so then catalase tests negative. Spore of Clostridium mostly oval shape, not a circular shape. Oval shape mostly. They can synthesize so many organic acid, alcohol, or exotoxin. It causes wound infection when you get cut by, well, whatever, knife or some animal scratch or bite. Well, animal bite you, dog bite you instead of rabies. Clostridium need to be concerned about anaerobic if the bite wound is very deep. So once animal bite or knife cut into very deep tissue, when knife pull it out, tissue will come to close each other, and that area is an aerobic area. So if knife or whatever, we believe rusty knife, rusty nail that pinch into your skin might contain so many spores of Clostridium. But we know now, even the knife that you just clean, cut you can cause infection of Clostridium also. As I said, the cleaning in the proper way cannot deal with bacteria that produce endospore. Okay. There are 4 bacteria. Well, I probably need to add some more. There are 4 bacteria that can be that kind of important in the genus Clostridium. That actually there are more, but 4 of these are important. One is Clostridium tetani. Second one is Clostridium botulinum. The third one is Clostridium perfringens and Clostridium difficile. For 4 of them. Let's start from tetani. So Clostridium tetani has, well, this bacteria is very important and people spend lots of time for study. The general characteristic of tetani is anaerobic, strictly anaerobic. People sequence genome of Clostridium tetani and analyze genome structure, and we found this is the model of tetani cells. Okay, the purple and the orange on the gray area represents cell wall of Clostridium. Okay, the purple here is a channel protein, the ion channel, sodium channel. This is sympod, bring 2 things come together. This is antipod, 1 thing out, 1 thing in. So there are so many channel types on the membrane. And if you consider types of the channel, most of them are channel for pooling amino acid, not sugar. So Clostridium can be 2 different types. One type is saccharolytic Clostridia. Another type is proteolytic Clostridia. It depends on species. Okay, this is general characteristic. Saccharolytic Clostridia is Clostridium that use sugar and convert sugar to be so many type of acid. Okay, but proteolytic Clostridia, they consume amino acid and convert amino acid to be something else, some intermediate. Okay, Clostridium tetani is one that is proteolytic Clostridia. They consume a lot of amino acid. They get energy, I mean ATP from metabolite amino acid. Okay, and one key of the the pathway of tetanol is this one is Strickland pathway. Strickland pathway is the way that Clostridium consume alanine and two molecules of glycine together. Well, then they change alanine to be pyruvate and ammonia because they move ammonia out, okay? They move ammonia out of this and convert to acetyl coenzyme A. And finally, the metabolite of this get one molecule of acetate by this way and two molecules of acetate in this way. Finally, you get three acetate. This is acid, okay? And carbon dioxide and three ammonia molecule with three ATP. Once these pathway happen over and over and over, the byproduct of pathway is ammonia. That can explain why Clostridium tetani, when they infect to your skin, they cause tetanus. Tetanus in Thai we call batayak. And when you infect wound, they destroy your muscle because they metabolize the amino acid and generate bad smell of ammonia. Okay, the wound get rotty and then bad smell. Okay, that's the characteristic of Clostridium tetani. So cell, for bacterial cell, Clostridium is gram positive. Okay, but if you keep culture longer than a day, and then you stain the old culture, you probably get gram negative. Clostridium can be gram variation and change from positive to negative. Okay, this is characteristic. Spore is very round, okay, and present in the terminal, the edge of the cell. So in Clostridium group, position of spore can be one of the key to help you identify species, particularly among four of these Clostridium. This one is super easy. When you perform gram staining, you can see spore easily. Spore happen so easy. And position of spore present at the terminal. The cells keep pattern of drumstick, mighty claw. Okay. This is obligate and arrow. Need carbon dioxide fill in the chamber for growing. Can grow perfectly in 37 degrees Celsius. When the cell grow, they produce a film like this. This is one inoculate of Clostridium tetani cells grow in the film-like structure. Hemolysis can happen, but they start from alpha hemolysis. And if you keep the time, then it shows beta hemolysis later. So key point of Clostridium is they produce a toxin. And toxin for Clostridium is AB type toxin. I will not go to detail, but just concept. The toxin of Clostridium named tetanospasmin. Okay, tetanospasmin is exotoxin. Exotoxin, that means toxin they produce and secrete out from the cells. Okay, let's see this picture. This is neuron, axon bulb of one neuron, and this is another axon of another neuron. This is motor neuron because they send the nerve impulse to activate muscle. Okay, normally the motor neuron will send neurotransmitter here to muscle. The neurotransmitter here is acetylcholine. Okay, but if they send acetylcholine the inhibitory interneuron, I mean this one, will send another neurotransmitter. It is glycine and GABA, depend. Okay, when glycine and GABA bind to the receptor, it blocks the nerve impulse in the motor neuron. So then they stop releasing acetylcholine. That means the muscle relax. But if they stop releasing neurotransmitter here, acetylcholine release, muscle spasm, muscle contraction. This is normal state, okay? But the toxin, the tetanospasmin, go to bind the inhibitory interneuron here to block the interneuron cannot release glycine. So once interneuron cannot release glycine, they cannot stop the impulse from motor neuron. The impulse sent here, they keep releasing acetylcholine. Okay, muscle spasm. Spasm is mean muscle contraction and cannot be relaxed. They spasm all the time. Okay, so people who get infected and get tetanus, they might have 3 different symptoms. Trismus is very not mild, but in the one way is if they got not so much toxin, the first is lockjaw, ขากรรไกรค้าง okay, so doctor can measure the length of teeth because you particularly happen by, well, in your face you have a big muscle that pulling your lower jaw up and down. That action, the toxin action in that muscle to make the muscle spasm. So muscle spasm, they pull your lower jaw up. You cannot open the mouth. You cannot move your lower jaw, cannot chew, and you kind of the top and the low part of the jaw spasm a lot, and in some severe case they damage the tooth. Tetanic trait toxin happen by so many muscle on the face, or maybe some part of body, but mostly they happen on face. Okay, face muscle spasm, so your face shape is changed. Some muscle can contract a lot. And the final thing is opistotonus. Opistotonus is mean all muscle in your body spasm, get contraction. All muscle is mean all muscle in the cardio muscle, the smooth muscle, or every muscle. So your lung cannot breathe, your gut cannot squeezing, your intestine, everything, then you die. Okay, that's the tetanus. Prevention. If you get tetanus, so we probably use tetanus immunoglobulin. Is the same as snake venom. We cannot waiting until you get antibody, okay, because you probably die before. The prevention one is vaccine. Like we know tetanus action by toxin mostly, right? So we combine toxin together in the DTP. Still remember what is DTP? Yesterday you learned
diphtheria and tetanus pertussis, right?
Yes, diphtheria, tetanus, and pertussis. Pertussis you learn next lecture. That's all that you should know about the Clostridium tetani. The second one, Clostridium botulinum. Oh, sorry, this is wrong. Botulinum here. You see this picture, spore is oval, not round like tetanus, and spore is not present at the tip but slightly moving inside the middle. We call subterminal. Okay, sometimes you probably see the free spore because the vegetable cells lies already. You can see spore inside the cells, free spore like this. This is pattern of Clostridium botulinum. Botulinum is gram positive, but when you stain, you probably see gram negative. This is the characteristic. Okay, botulinum can produce beta hemolysis or non-beta hemolysis, depend on strain. Okay, colony is quite big, right? Botulinum is happen contaminated by spore or vegetative cells. Normally, vegetative cell die already, but spore still remain in the preserved food. In some country, people making their own pickled cucumber, kimchi, or in Thailand, we make a bamboo shoot. Right? We preserve bamboo shoot. And you probably hear some news some year that people die from eating bamboo shoot contamination with Clostridium botulinum. Okay. Canned food, preserved food, that's the target. Once you close the jar or close the lid of the can, you probably the food factory will fill some juice, some water, some soup, fill in the can until the top, and that's become an aerobic condition. That's very nice for botulinum. So botulinum hatching from spore and produce so many, well, they use carbohydrate, they use protein inside that, produce so many acid and gas. You probably detect the can with the wrong shape, the can swollen, then cannot eat. One news, this is from the true story, a housemate, she made her own pickled cucumber, okay, and she feel like it probably rot, or the in Europe or in America, when you buy pickled cucumber, the can will be you probably see two type of how can I say, the lid. The lid can move into two pieces. That for detect the botulinum. If they produce gas, the lid will show up like, and you can press it and you hear the sound. Flip, 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 flip. So she feel like this is rot. But the thing she did is she opened it and she used her own finger, dip into the cucumber, pickled cucumber juice, and she tests that. And she feel like it is normal, not rot at all. Then she cooked pickled cucumber to everyone in the family. The day after tomorrow, the day after that day, she died. But the other member still okay, but she died. That because she dipped and she get infected by Clostridium botulinum without eating food that heated. If the heat can destroy the toxin, this is the thing, and it's a true story in the other country. So police thought that she has been murdered, but actually it's not by Clostridium. So toxin produced from this bacteria called botulinum toxin. Okay, botulinum toxin, also neurotoxin, same with the tetanus, because the action at the axon. Okay, this is motor neuron, same with the previous picture. The motor neuron release acetylcholine to activate muscle and get contraction. Botulinum inhibit releasing of acetylcholine because this is the inside of the axon. Acetylcholine stay in the compartment in vesicle. The vesicle need to move to fuse with the membrane here and release acetylcholine to synapse area. Okay, they use protein on vesicle linked with another protein on the membrane. So botulinum destroy the protein on the membrane. That's why the vesicle protein cannot bind to the membrane protein and then cannot make the membrane fusion. So acetylcholine never release out of the axon. So once acetylcholine never release out, muscle never contract. Muscle never contract is mean muscle paralyzed. Okay, the disease are opposite situation with tetanus. So people die in severe case. People die from paralysis. So all muscle in your body in a severe case cannot contract. You cannot contract your heart cannot pump blood, lung cannot contract, the gut cannot contract, intestine cannot contract, so you completely die. Okay, but also same, botulinum toxin is one of the very serious, together with tetanus toxin, botulinum toxin, anthrax toxin. So three of these are used as bioterrorism agent. Okay, this is very serious. And another application of botulinum toxin just for people who get Parkinson's disease, they're shaking a lot. So applies toxin to stop sending or stop releasing acetylcholine, the muscle will not shake. So it helps when people get severe muscle shaking, particularly on the face. Okay, and also cosmetic use botulinum in terms of Botox. But in the very, very low dose, otherwise you paralyze, your face paralyzed. Okay. This one, Clostridium perfringens. Perfringen is not a food poisoning. Perfringen is cells shorter than tetanine or botulinum. Cells very short, and you probably see the cells staining gram positive mixed with gram negative because they kind of gram variable. Okay, you can see perfringen in a single cell or in pair. That's the characteristic of perfringen. Can be pair or single. Spore present in subterminal, but you cannot see spore easily. Spore is not produced easily on culture, even in vivo. Okay, this is characteristic of perfringen. Perfringen cause gas gangrene when you get the wound. Wound happen by trauma, accident, or operation. If the doctor or the healthcare section not take it very serious, then perfringen will infected. Perfringen can consume sulfur. That make the wounds turn black. Okay, and also can cause severe food poisoning too. But this one is dominant of perfringen is gas gangrene. Okay, gas gangrene is the wound is inflamed and swollen. Inside the swelling, they have bad smell gas inside. Okay. The last one, Clostridium difficile. Difficile, the cell is quite long, okay, compared to perfingen. Spore present in subterminal and terminal. It depends, but you cannot see spore easily. Also, same. If you not see spore easily, and you see the gram variation, you probably need to think about long cells or short cells. Okay, and difficile itself is non-hemolysis on blood agar. The colony shape is not help much for identifying species. Okay, so this is very important. Normally, C. difficile present in everyone as normal flora in colon, but in the very low number. The reason that make C. difficile, or some people call C. diff, increased population because that one get antibiotic. Well, antibiotic get inside, they kill most of bacteria, most good bacteria, and that bacteria, some of them that die out. It make the colon lost balance of population. So C. difficile overgrowth. When it overgrowth, they can cause pseudomembranous colitis. Here, the pseudomembranous colitis is colony of C. difficile in the colon. It look like the another membrane, and you get severe diarrhea by AB type toxin also. Okay.
อาจารย์ค่ะ
ครับ
Could you repeat the slide 27? I think that internet was a bit cut off. ค่ะ
This
Yeah, when you talked about the antibiotic of the bacteria.
Okay, so antibiotic bacteria wipe out when people who get antibiotic a lot, overuse antibiotic. Do you think which group of patient overuse antibiotic? So cancerous, right? People who get cancer, they need to be take over, or people who get HIV, they need to protect themselves from antibiotic. And antibiotic kill the good bacteria. Okay, once the good bacteria die out, C. difficile overgrowth because they lost balance between bacterial population. When C. difficile grow, they produce more toxin. When the toxin producing, they cause pseudomembranous colitis. Colitis means inflammation in colon, and pseudomembranous is mean like they grow up like a pseudo abscess, the white area inside the colon, like this picture. That's come from the action of toxin. That's the thing. Okay, I will give you some idea a little bit about the toxin. We will have some more time to talk about toxin again, but since you learn bacteria, you probably hear the word toxin so almost every lecture, right? But toxin can be so many types. You need to go back to review yourself. In the very beginning, we said the cytotoxin, the toxins that can destroy a host cell by making pore, something like that. This cytotoxin. Every toxin that secrete out of the cell, we call exotoxin. Okay, they secrete out of the cells. Cytotoxin harmful for the cells, make cell lysis. AB type toxin that refer to structure of toxin, can be structure A and B bind together, and then one toxin action, they split A and B together, and A do one function, B do one function, like this AB toxin. You learn a lot more in the AB toxin, in E. coli, in Pseudomonas, in Salmonella. There's also produced, one bacteria can produce so many types of toxin, okay? Botulinum toxin, tetanus toxin, tetanosporin, this is tetanus toxin, AB type. Corynebacterium diphtheria also produce AB type toxin, diphtheria toxin, okay? There are so many toxins like this. All of these are categorized to be exotoxin, okay? Something else. Oh, the other type of exotoxin that I forget to mention is erythrogenic toxin. Okay, in Streptococcus, also toxic shock syndrome toxin. That's another toxin. So they are all exotoxin. Another toxin that you probably need to know and we will learn from the next lecture, we call endotoxin. Endotoxin is lipopolysaccharide that found on the outer membrane of gram-negative bacteria. So then we never talk about endotoxin in gram-positive, okay? So gram-positive process exotoxin. Gram-negative can do both, okay? Exotoxin mostly protein. So then heat lay-by. If you heat the food or contaminate thing, particularly food, toxin destroy. You probably save, okay? But endotoxin are different. Endotoxin is lipid, okay? Even you heat it 100 degrees Celsius, endotoxin still remain and can function because it is lipid and carbohydrate, okay? That's the thing. This table, you probably go to study yourself the property between exotoxin and endotoxin, how different. Okay, summary. Gram-positive rot can be endospore. You learn Bacillus clostridium, okay? Non-endospore forming. You learn regular shape is Listeria, Discipulothrix, okay? Irregular shape, non-acid fast. You learn Corynebacterium, Propionibacterium, okay? Acid fast, we talk about Mycobacterium. Of filamentous branching cells bacteria, we talk about Actinomyces and Nocardia. That's the big picture of gram-positive, okay? Do you have anything to ask?